we'll just wait a couple minutes to start while people still sign in. So Nancy, you can see my screen now, right? The shared screen looks okay. Nancy, can you hear me? Okay, great. Thanks. Let me just one more thing. Okay, I'll start recording.
Recording in progress.
Okay. All right, everyone. So welcome back. So we before I start, I just want to say we very much enjoyed all the questions you guys asked last time. So it really helped us to tailor the course and then get the interaction with you guys. So please do still ask your questions into the chat. If by any chance any of your question wasn't answered, and we'll try to email you. If you didn't receive any email, you feel free to just email us. Any questions you still have. Okay, so today we'll start with what we left as one example to kind of demonstrate the deep learning in omics data. So this example is to predict the interaction between T-cell receptor and HLA. So it's continue on the immune aspect of the omics data. And the main goal here is to talk about the hyperparameter selection. Okay. And the example of the code is already in the GitHub. So we start talking a little bit background of the immune response. The COVID make us all probably learn a lot more about the immune response. So like any infection, common cold or flu, the immune response usually have two phases. The first phase is innate immune response. As soon as you got the infection, your immune system will fight against those infections. But it's not that specific, and then it's not that strong. So usually at the beginning, you start to get the symptom and then feeling cold, feeling bad about that. And then after 6 or 8 days, the adaptive immune system can kick in. Basically, the immune system take time to learn what is the virus, and then they can basically start more specific attack. So that including T cells and the B cells. B cell is T cell receptor is the thing we're going to talk about. B cell receptor is basically the antibody we talk about. The B cell receptor can be on top of the B cells. It could be also just separate the antibody in the blood can or in your nose, your throat can fight against the virus. Okay, so just remind you, this is something we touched a little bit last time. So that's the way the T cell recognize the infected cells. So you got the T cell here, and then you got infected cell here, right? So basically T cell will kill the cell. It does so by recognizing the peptide or the antigen presented by the cell. If the cell is infected by a coronavirus, it will present some of the protein from the virus, and the T cell can look at that and say that's not our own protein. So it will attack the cell, basically kill the cells. And then here, last time what we studied is the question, what's the association between MHC or HLA with the peptides? And now we want to study the association between the HLA and the TCR, basically the MHC or HLA here with the TCR. before that, I just want to give a little bit background, why this is an interesting problem. So we can actually use the T-cell receptor data to predict the infection history. So you think about in a case control study design, some cases they are infected, and it doesn't have to be like current infection. It could be an infection like months or even years ago. So we can look at the infection history. And then we collect the TCR data from each individual. So the TCR data is kind of like SNP genotype data. You have a big matrix. Each row is one TCR, and then the data is just 0, 1, whether the person has this TCR or not. And then you can look at the association between each TCR and the disease. So that's just like a GWAS, right? And then you can predict the disease status using a weighted summation of TCRs. They are associated with case control. If you're familiar with the genetic association or polygenic risk score, you will recognize this is exactly the same thing. In genetic studies, we often do that. We find the SNP associated with a trait, and then we do a weighted summation of the SNP genotype, and then we use that to predict the complex traits. And then also, this approach is pretty powerful, but there's an important confounding factor, which is the HLA. Okay, so this is a figure using the data generated from earlier publication to study the TCR data to predict the exposure to CMV1 virus. It's pretty popular virus. Half of us have it. We just don't know it because it doesn't bother us. The immune system can take care of that. It only become a problem if somewhat a person is immune compromised. For example, if one person goes through the, say, bone marrow transplant, the immune system will be basically removed before the transplant. And then the person has to be tested for the virus, and then otherwise the virus may cause very serious outcome. And then, so the plot can be read like this. So each point is one individual. Okay, and then the x-axis is how many unique TCRs you measure in that individual. And the y-axis is the number of disease-associated TCRs. So those are the TCRs, they somewhat can recognize the virus protein. We identify them from association studies. And the color is the individual who have the virus, who do not have the virus. And this is the training data, this is the testing data. So you can see actually the signal is very strong. If you consider this is like a GWAS problem, like you can predict the trait very accurately. I mean, training is like perfect, but in testing, you can see there's still a good separation. And then the way we select what TCR associated with the CMV is based on a p-value threshold. And now if we consider, if we use a different p-value threshold, then look at the AOC, the area under the area under the curve, and then that tell you overall how accurate the classification. And so the all subtract, the right one is if you look at all individuals. So the p-value we use here, I think it's 0.001, so actually corresponding to a point here. So if you just look at the right curve, that's kind of as expected. If you have a more liberal p-value, p-value 0.01, you got more disease-associated TCR, and then the AOC a little bit lower, it goes high, and then if the p-value is too high, you don't got too many TCR left, and then it goes down. So that's the red. However, if you're conditional on the individuals with a particular HLA allele, for example, if we look at the purple one, if we look at all the individuals who have this particular HLA allele, you can look at the AOC, it becomes much higher. So this is saying, the association between TCR and the disease, it depends on the HLA allele, because that's basically the underlying mechanism. The TCR recognize the complex of the HLA and the peptides. So maybe the peptides or the protein from the virus is more likely to be presented by this HLA, and then this HLA is more likely to be recognized by the TCR. So you can see for some HLA, you got much better prediction. And it's also possible for some other HLA, the prediction is worse when you condition on a particular HLA. So I want to use this example to show you, first, this TCR is very informative data. They really can, it's easy to get. From one drop of blood, you can measure the TCR, and then you can basically understand the infection history of many infectious disease. So that also gave us a motivation that why we want to study the association between HLA and the TCR is a meaningful question, because we see the HLA play really important role. For example, we can try to account for HLA in the association analysis. And basically, we want to study which TCR is associated with the disease. We can do that by conditional on the HLA. And it's challenging problem because there are more than 15,000 HLA alleles in the human population. You cannot do a conditional analysis, conditional on each one of the 15,000. You need like tens of millions of samples. And then many HLA alleles are pretty rare in the population. And so here we want to have a little bit more general approach that we want to say first study the HLA and the TCR using a neural network model, and then we want to predict whether they are associated or not. So this information can be helpful for many downstream analysis. So here we just use the sequence as input, and then so that if we have there are 15,000 HLA allele out there. If we only use like 1,000 HLA allele to train our model, and then we got a new HLA allele, but we know it's sequence, so we can still plug into our model to make prediction. And then here, the training data we got is the positive HLA TCR pair, they associate with each other from the previous study. Basically, in that study, you observe the HLA, you observe the TCR, and then you can just calculate, a facial exact test and then find the associated pairs. And the negative pair is just randomly sampled once. And then now back to our neural network. At this point, we can also say, this is also an example where the neural network can be better than other methods. And the rationale is same as the previous approach when we talk about the HLA and the peptides. Basically, there can be some interactions. So we can use the neural network to capture that information. And the neural network is similar to the one we talked before. You got the sequence from HLA, so that's 34. Here we actually use more amino acids, it's 40 amino acids. And then the TCR sequence is variable, but you probably can skip the detail. There are like different parts, CDR1, CDR2, CDR3. And then we encode them, we can concatenate them, and then we can also use the CDR3 is the part which is more variable. It's generated by somatic mutation within T cells. So it's actually different from every T cells. So we also can use the length here as an input. And then we can build a neural network and make prediction. And so the example code is in the repository. The neural network is nothing new. It's very similar to what we have before. And the thing I want to illustrate is one thing is how to choose the hyperparameter, and then they also gave an example, actually different type of neural network. So we tried different architectures. So this is one simple one. So you have the TCR part, you have the sequence and the length, and then you have the HLA part, and then you put them together. And then this dense layers, it just means fully connected layer. All the nodes in layer 1 connected to all the nodes in layer 2. So that's a dense layer. And then you got a prediction. Okay, so next one, you can actually add a little bit layer to somewhat encoding the sequence first, right? So you have the TCR, you encode them. You have HLA, you try to encode them, and then you put them together. So you try to encode them separately. Give a chance for them to learn what's the better representation. And you can also add a convolutional layer here. So the convolutional is, it also comes with a max pooling. So basically, maybe the information I want to learn is now position specific. Maybe if I have certain sequence in the first position or the second position, they can have the same consequence. So I can use convolution max pooling to give that flexibility. And also add two convolution layer, and also we can add LSTM. So that's recurrent neural network. That's something we didn't talk. I just want to give a very brief overview here. Recurrent neural network is pretty popular in, say, natural language processing. So it's particularly designed for a sequence of data. You know, when I talk, the recurrent neural network can try to predict what is the next word I'm going to say based on all the things I already talked. So you can understand the recurrent neural network as something like a fancy version of a hidden Markov model. You know, hidden Markov model, hidden Markov model is probably the most successful model for natural language processing before the neural network. So the hidden Markov model try to learn what's the current state condition on the last time point, right? Condition on the last time point is independent with everything before that. So that's the Markov assumption make the hidden Markov model computationally doable. But in the language processing, for example, you know, we talk about this one. The clouds are in the sky. If I want to predict the word sky, you know, this sentence is enough. You know, the hidden Markov model can be modified. You can depend on probably a couple time points ahead. You know, the clouds are in the sky, so that's easy to guess. But if you know, I have a longer sentence, I grew up in France and I speak French. So if there's something else there, so you need a longer history to guess what is current state. And that's where, you know, the hidden Markov model will have a problem, because if you have too long the history, the hidden Markov model becomes computationally infeasible. So that's where the recurrent neural network can do pretty well. And the network we use is called LSTM. So that's a very popular recurrent neural network. It tries to solve a problem for recurrent neural network is actually when the history becomes longer, the recurrent neural network become less effective. It's more the some technical region, the gradient will shrink a lot as time go on. So here, the LSTM is a pretty complicated graph. So I try to go through this briefly. There are actually also many materials out there. This is one of the figures I find is most clear. The basic idea is, you know, previous one you see here, you have the data, you know, that's like the word I'm talking, and then the sound you hear, and then you try to infer what I'm talking, the hidden state. There's A is like a box, so the neural network. And now here, in addition to the hidden state and the input, we introduce another is a cell state. So the cell state actually goes from one time point to next point more directly. So it's like build a more like direct connection across the time, so that you can make the gradient, when you do the stochastic gradient descent, you can make the gradient easy to control. And then operationally, it's more complicated, like you got the input, you got the hidden state. And then, so here, like this is kind of dimensional data, you concatenate them. And then there is a forgot state. So basically in the cell state, how much you forget. And then addition, so you also try to combine them and then try to add a little bit more information. I should have annotated here, S and T is a different activation function. But the detail is probably not that important. You know, you just know they try to modify the cell state. You forget something, you add something, and then you also combine the hidden state and the input to get a new hidden state, right? Combining hidden state and the input to get a new hidden state, that's what standard recurrent neural network do. So what LSTM is basically give a cellular state, a cell state. With that, we look at the result. We consider 6 different model architectures, and then there are also many other hyperparameters. So we consider the way we encoding the amino acid. We say one-hot encoding, 0, 1, but there are alternative options. You can use some biochemical property of the amino acid to encode them. And then there are also the way we use the V gene in the TCR. There are TCR1, CDR1, CDR2, they are part of V gene. So there are different ways to include them. And then the layer configuration, how many nodes or how many neurons per layer, and then the dropout rate. Okay, so we put them together. Altogether, we got 720 instance, right? They could be different models or different hyperparameters. And then we look at the AOC. We classify the HLA TCR association. And then you can see, basically this is a histogram of 720 instances. The vast majority of them is around 0.65 to 0.8. There's very small proportion around 0.5. As AOC, 0.5 means you're doing nothing, right? It's not doing anything at all, but most of them is actually pretty good. So the thing I want to emphasize is really, the hyperparameter tuning is hard. I mean, it's take time and then take experience. But one thing is really important is there is enough information in the data. The hyperparameter tuning may get you, say, from 0.7 to 0.8. I mean, it's big increase. But another probably more important thing is, you got enough information here, and then the network is overall is pretty robust. You don't want this go the other way. Like the vast majority is 0.5. There's only a couple of them with 0.7, 0.8. I think that would be more dangerous situation. You want to say, why you change the tuning can change the performance so much. So there's a quick summary. There are, to do the hyperparameter tuning it's a hard task, and the space of model architecture hyperparameter can be very huge. So you could do this manually. You could there is a specific method, the Bayesian optimization. Basically, you try to choose the hyperparameter adaptively, and there's also other method like hyperband. So that's like try to allocate the resource. Basically, I have this hyperparameter set up. I just go a few epochs and then decide is it good or not. So I don't spend too much resource on that. So there are different ways to do that, and there's a sophisticated package you can use. But I think it's really, at the beginning for many new problems, like for many omic problems, people may not apply deep learning on that. The first step is really to ask, are there enough signal noise ratio? Are we finding anything from the neural network? Is it really doing better than standard approach? So I think it's the lesson I want to emphasize here is really there should be high enough signal noise ratio. So the result is not highly sensitive to the hyperparameters. Okay, I think I can stop here. I'll switch with Nancy. I will start monitor the chat and answer questions. I will stop sharing here.
Okay, hi everyone. Let me share my screen. Put my chat here. Okay, so yeah, so I'm going to start on the next module for this course, and so for most of, I think the rest of this lecture, we're going to be talking about unsupervised learning, mostly with autoencoders and variational autoencoders. So here are the learning goals. I'm going to start with just more details on how the autoencoder works and introduce the variational autoencoder, which is a very useful variation of this tool. And then I'm going to go into detail in two examples. The first example is from single-cell transcriptomics. I'm going to talk about how the autoencoder and the variational autoencoder allows us to do denoising and batch correction and data integration in single-cell transcriptomics. So here, the word that we oftentimes use is transfer learning, and you see that a lot in deep learning, and you'll see how transfer learning with deep learning works in single-cell genomic data. And then the second example is a recent study where the variational autoencoder is applied in a very creative way to predict the pathogenicity of mutations within the protein sequence. So we're going to see how that works and how especially the variational autoencoder allows you to have a beautiful solution to that problem. Okay, so as we're going through these examples, what I want you to ask in the back of your mind is what makes these problems really suitable for deep learning? Okay, and we're going to have discussion about this topic after each example. Okay, so first, autoencoders and variational autoencoders. So autoencoders, as you know, are a tool for unsupervised learning. What we mean by that, we mean we have an X matrix, row by column. Each row, say, is an image. Each column is a pixel. And there are no labels, right? So you don't know which image is a cat, which image is a dog, which is a car, which is a boat. But yet, you still want to learn something about the underlying structure of the X. So the way an autoencoder works is it maps X to a low-dimensional latent space or an encoding Z, and then it does this mapping so that the Z has to be good enough because it's low-dimensional. You have high-dimensional data. It needs to be good enough to reconstruct Y through the decoder. X through the decoder, and then the loss function that you use to train the autoencoder is just I'll show you different versions of the loss function, but the vanilla autoencoder is just a Euclidean loss, the squared error norm. Okay. So, and then you see here, I just want to write this out so everybody is on the same page. For example, there's two internal nodes in the bottleneck layer, and node J has basically, actually, I wrote this wrong. This F should be in the outside. Sorry. F. So this is a very important detail. So this F is the activation function, and then so you basically take all the input layer nodes, you multiply them by the first layer weights, and then you sum them up and you feed them into the activation function. So the simplest autoencoder is the PCA, which I think everybody is familiar with, right? So in the PCA, you want to map X to a linear, you want to have the best linear approximation to your X. So you want to project X onto the space of V, right? V is your loadings matrix. And then U here is your scores, your linear PCA embedding. Right, so U is X times V. V is optimized over the space of orthonormal matrices, right? So the relationship between the PCA and this simple linear autoencoder is that your F function needs to be just identity, and then your V here would be this set of weights here. The U would be the linear embedding would be the two internal nodes. Here we're just mapping it to 2 dimensions. And then the V transpose will be these weights. But most autoencoders are multi-layer neural networks where your encoder and coders have more than one layer, and your activation functions will be nonlinear. And in that case, what you're doing is you're projecting your data onto the best fitting nonlinear manifold. Okay, so the basic vanilla autoencoder is actually already a very useful tool, but there are some caveats, right? So remember, when we are optimizing this thing, we're just saying we want the best reconstruction of X. There's nothing in the optimization in the algorithm that says this latent space Z has to make sense. And in fact, you can show that if you have enough hidden layers and enough nodes in the hidden layers, you can just map any high dimensional data into a single bottleneck node. So as a latent space of dimension 1, and you will be able to reconstruct your data with perfect accuracy. So that will be overfitting the data, right? But in that case, your latent space will have no good interpretation. So what we would like to do is to interpret the latent space. We would like to have, for example, Z values, embeddings that are close to each other to be mapping to similar X's. right? So that's what we call continuity. We will also like to be able to sample from the latent space, and you will see examples in single cell literature where that makes a lot of sense. And then you want to do algebra on Z. Say, for example, you want to like subtract to do linear shifts in Z, and I will show you examples in single cell data where we want to do that. And also, in some cases, you may want to say, for example, I want to find an embedding of my data that is orthogonal to some covariate. I want to remove or account for the covariate effect in my embedding. And with the vanilla autoencoder, even if you just add a node B to the internal layer and then say you want to, you have to do more. You have to fiddle with this in order to achieve that goal. Okay, so I'm going to start with the variational autoencoder, which is a particularly useful version of autoencoders. And the variational autoencoder starts with encoding layers. And what is different here is that in the bottleneck layer, what you learn is the mean and standard deviation vectors, which you then use to sample Z. Okay, so instead of learning a fixed function Z of X, okay, you learn a random distribution, a conditional distribution of Z given X. And then from that Z, you want to decode back your X, and then you want to assess how well X hat approximates X. So I think most of you are statisticians. You probably heard of this Bayesian variational inference route of deriving the variational autoencoder. I don't have time to go through that in this lecture, but I think we gain a lot of insights by just looking at the loss function that you use to optimize the variational autoencoder. So the loss function has this reconstruction loss, which is what we had in the previous slide, is how well X hat approximates X. But it also has this KL divergence term that compares your conditional distribution of Z given X, which is this, we assume that it is normal, because what we do is we sample a normal zero identity, a C vector, and then we just have Z equal to mu plus diagonal sigma times C. So we compare that distribution to the standard normal Gaussian identity covariance distribution. And we want that loss term to be small as well. The KL divergence penalty to be small. Okay, so if you know about Bayesian variational inference, this is equivalent to the negative of the ELBO, the evidence lower bound. Okay, but if you don't know about it, don't worry about it. Okay, so what this does is it results in a latent space that is continuous, interpolable, and amenable to sampling. Now, why is that so? Well, for sampling, it's obvious, right? Because once you have estimated this mu and once you have estimated this Z, for example, you fit the weights of the encoder and decoder. What you could do is you can just sample Z from the prior, and then you can get X sample, have new sequences of X coming out of that. In terms of continuity, if you imagine, adding this noise here gives some robustness to the system, right? It's saying that, let's say you start with an image of a cat, and then you map it to, say, the mu vector, which is the mean. If you deviate from the mu because you have to, you add a noise to it, it still needs to give you back an image that looks kind of similar to the cat you begin with. So adding this noise means that, and this Euclidean noise, it means this normal noise, it forces the latent space to be continuous in the sense that you have to be able to fudge around the initial mapping value and still get back something that is similar to what you had before. So here is just a toy cartoon example. Let's say you're working with handwritten digits. On the left is the kind of embedding that you would get if you were to use the loss function that is just a reconstruction term, not the penalty. And on the right is what you get if you do have the penalty term added. So what because if you don't penalize for the KL divergence, there is nothing that says that the sigma values, these sigma values, shouldn't shrink to 0. Actually, it is beneficial for the loss, for minimizing the loss, for them to shrink to 0, because that would mean that you're not adding any noise, right? It's better to not add noise. So if you don't have that loss term, then it's essentially the same as not adding that sampling step. And then with this loss is saying that your reconstructed Z conditional distributions need to look somewhat similar to the normal 0 identity distribution. The means cannot just drift off to infinity, and the standard deviations cannot shrink to 0. So what that means is that your points are going to run, the distributions are your balls are going to run into each other, intuitively. And then, so what that means that you can sample from this space. For example, if you have a point here, it's close to a 7, so it should be a 7. And maybe, you can say that it is, it's in the direction of a 1, so it should be more narrow, right? This is just a toy example, but you can get the intuition, right? So if it's between a 7 and 6, maybe it should look something like the middle of those two values, and etc. So that was a cartoon, but here is an actual data. Here is an actual VAE embedding of handwritten digits in 2 dimensions. And you see that it makes sense in the sense that, you can go from a 9, slowly it morphs to a 1, it morphs to a 0 in the other dimension, et cetera. This is only 2 dimensions. So you can imagine if you have like 3 or 4 or 5 dimensions, your data, your latent dimensions will make some sense. Okay, so I know that's a very short introduction, but I hope that gives you more familiarity with autoencoders. And then so let's think about, next I'm going to skip into single cell transcriptomics. I'm going to give you some background on this field, and then we can look at how autoencoders are applied. Okay, so why do we want to do single cell experiments? The reason is because each of our body's tissues is actually made up of very heterogeneous mixtures of cells. So our body is actually a tightly knit community of cells working together so our tissues can function. Okay, so we would like to tease apart what kind of cells are in each tissue and what happens to these cells, say, when we get sick. And then, the cells you can imagine are sort of like little autonomous units of our bodies. They're not just, each cell has its own DNA, which is the same across cells, but it makes its own decisions about what genes to express. So, for each cell, it expresses DNA into RNA, and then the RNA gets translated into proteins, and most of the job is done by proteins. But in single cell RNA sequencing, we sort of extract the cells and sequence the RNA, this intermediate, and that gives us a sense about the state of the cell. Okay, so it's been more than 10 years since the first single cell sequencing experiment was done way back in 2009. And that experiment was on a very small number of cells, I think only 6 or something like that. But now it's routine to have experiments where hundreds of thousands and millions of cells are collected. Okay, so, but then with the rise in the number of cells that are collecting each study, we have a limited budget, so we cannot sequence very deep in each cell. A typical experiment nowadays would sequence around 5,000 UMIs per cell. Okay, and that is a sample from the total RNA pool in each cell. So you can think about each cell being like a bag of RNA, right? How much RNA is in there? There's about 50,000 to 200,000 transcripts per cell. So you're taking a small sample in order to sort of have it be hopefully a representative sample for that cell. Okay, and then obviously, so if the larger the sample, the higher the coverage of sequencing for each cell, the better the data you're going to get. And with better data, you will be able to tell apart more subtle differences in cell state, right? So in this picture, you have on the X a very low coverage sequencing experiment, okay? And on the right, as you move to the right, is a high coverage sequencing experiment. Go out. And then So you see that when you have a low coverage experiment, your cell types sort of all merge into one big glob. And then when you have high coverage experiment, they separate out in, say, a low dimensional PCA embedding. Okay, so we're going to look at several different problems in RNA, single-cell RNA-seq analysis, one of which is data denoising. So what that means is that you're going to take the data you have and then try to recover what you would have seen if you were to sequence this data at much higher coverage. Okay, so data denoising means to try to extract, to remove some of the noise due to low coverage. Okay, so here is an actual example of a data set where we have 900 TBMC cells, meaning the immune cells in peripheral blood. And what you're seeing is a UMAP. UMAP is a two-dimensional embedding of high-dimensional data that preserves neighborhood structure. And the cells are colored by, in this case, we know the cell types. So we know that the purple are the B cells, the blue are the monocytes, and this big glob where everything's mixed together are the T cells, okay? But it is important to know the difference between the different T cells because, for example, you have a naive T cell. This T cell is not doing anything. It doesn't have, T cells are the sort of the armies in our immune system. Wei was talking about how they attack pathogens and they gobble up cancer cells, right? So T cells that are naive doesn't have its guns out, okay? They are just not doing anything. And effector T cells are the T cells that are armed and ready to do the killing. So, it's pretty important that you will be able to tease apart these T cell subpopulations. So, you see that this is a low coverage experiment. I think only like less than 1,000 UMI per cell. But with denoising using an autoencoder-enabled transfer learning, you are able to tell apart these different T cell subpopulations. And I'll show you how that happens. Okay. Another question that we are often have to grapple with is batch effect correction. What that means is oftentimes you start out with a homogeneous cell population, like a tissue you're studying, and then you have two lab technicians doing the experiment. So those are the two batches. And then you look at the data and you try to project it into a low dimensions. The two data sets don't fall in the same space. Okay, so there are systematic differences between batches. Okay, so this problem, there's many statisticians working on this problem. It's a well-studied problem, even in the bulk sequencing days. But in single-cell sequencing, it is very, very a pervasive problem. Okay, and other than batch, there are other things there are other covariates that are often confounding our analysis. Say, for example, we're doing a study comparing healthy versus diseased individuals. And let's say we collect cells from each of these healthy and diseased individuals. And we want to see what cell types are specific to the disease, or, say T cells, what, T memory cells, what is different about T memory cells in healthy versus disease? Well, it might be that these healthy and disease, sometimes it's impossible to get well-matched cohorts. Okay, they have different sample level covariates like age and sex. And also there are cell level covariates like library size of the cells and mitochondrial content and other things related to technical aspects of how these cells are collected. So the question then is there a way we can sort of remove all these unwanted variations from our data? Okay, so that gives you sort of a primer on single cell data and problems related to it. So I'm going to go through 4 papers in this area. The first is on denoising, and I put that first because that sort of gives us a general gist of this problem. And then we're going to go into a more complex problem of batch correction. There, I'm going to talk about 2 papers, both using very similar variational autoencoders to do batch correction. And then we're going to talk about the prediction of unseen cell types, which is exciting, basically using variational autoencoders to predict what might happen to a cell type if you were to, say, stimulate it. Okay, so Okay, so first let's just go start with a statistical model, because we're statisticians, we like to think about models. Okay, so Y is our observed data matrix. You can think about it as a matrix with a row being a cell and a column being a gene. Okay, and Y is a noisy representation of some true, hypothetical true expression matrix, which is a true count of each gene in each cell. And we would like to recover X. That is the goal, okay? And X, because, each cell is, what makes a cell, right? The underlying processes, the underlying pathways that are happening in the cell. And we would like to capture these coordinated variations across genes that are induced by these pathways and shared processes through this underlying lambda, okay? So we think that X is generated by adding, so the lambda, so we think about X, the cells, as living in some high dimensional manifold in transcriptomic space, which is represented by lambda. But they're not exactly on this manifold because everything's random. The transcription is random, RNA transcription is random, RNA degradation is random. They're not going to be exactly on the manifold. So it's basically the shared variations across genes plus some idiosyncratic, unpredictable biological noise that is independent across genes. Okay, so our goal, well, you probably can guess it. We're going to use the autoencoder to try to learn lambda. But first, let's think about how we're going to how we're going to incorporate that how we're going to use lambda to try to get back our true expression matrix X. Okay, so first we need a noise model. And there has been quite a number of studies, and I think the consensus among many of them is that a Poisson model is good enough for most of what we want to do. So we're going to assume a Poisson model where each cell has its own library size, size factor multiplied to the XGC, and Y given XGC is Poisson LC XGC. Okay, so that, we're going to assume that. And then, so that's this arrow, this long arrow. And then, what is the distribution that relates X to lambda? We have no idea. We don't know I don't know what is a good distribution for that biological noise. So we're going to model that non-parametrically, okay? So we're going to assume that XGC given lambda, it has a mean, lambda GC is centered on lambda, but it has a variance that scales with lambda squared with a gene-specific scaling factor theta G. So theta G is our dispersion parameter. Okay, and that's quite an important assumption because we're assuming that basically there's this mean variance relationship with a dispersion parameter that depends only on the gene. Okay, so effectively, what this model with this technical noise model, Poisson, or you can use other technical noise models. So Poisson is easier. And then this gene-specific dispersion is that it gives you a very natural way of sort of building an estimate out of your machine learning derived lambda hat. So what we're going to do is I haven't told you how we do this yet, but we're going to take the observed raw data, the noisy data YGC, and we're going to use an autoencoder to learn a representation lambda hat. And from lambda hat, we hoped to get an estimate of X. And it turns out that in this simple model, your best estimate for X hat is just a weighted sum of your observed YGC and your prediction lambda hat. And the weights in that model is a combination of, is a function of your gene-specific predictability term, this dispersion term, your cell-specific library size, and your lambda hat. Okay, so what that means is that let's look at an example. Let's think of an extreme example where you have a very predictable gene. So this gene somehow is very well coordinated. It doesn't have much biological noise, but you're in a very low efficiency experiment. So your cells are very low sampled. In this case, what you would want to do is you would want to just shrink very heavily to your prediction, right? Because you really trust your prediction, but you don't trust your data. There's a lot of noise in this step, but not a lot of noise in this step. And that's exactly what happens, okay? So these are points, these circles are your actual data, the triangles are your estimates, and then the red line is your prediction, your lambda hat. So you're shrinking heavily to your lambda hat. And in the opposite scenario where you have a highly erratic gene, okay, cannot be predicted well, and you have a high efficiency experiment, okay, so LC is big. In that case, you probably just want to rely on your original observed data point and forget about lambda hat, okay? So the nice thing is that with a statistical model, you can actually dynamically, you don't have to worry about this step is taken care of. The W automatically adjusts. So, so finally, let's talk about the lambda, okay? In fact, I want to say that a lot of methods in the field just use the lambda for their denoised matrix. And we don't feel comfortable doing that because we feel that one of the reasons why we collect single cell data is to study the variation across cells within a cell type. And so we don't want to throw away that what was called biological quote unquote noise, this idiosyncratic variations, because it's been shown that many diseases and the aging process actually involves an enlargement of this noise. Okay, so for lambda, actually, I think in last lectures, people were asking the question, why do we use deep learning? Why can't we just use some vanilla traditional machine learning methods? In fact, that is what we started with in this application. We just started with an off-the-shelf GLM lasso to fit a lambda, used by enforcing some sparsity in these coefficients, and that's what we did, and that actually worked pretty well. We got, there's a lot of third-party benchmarks of the method, and it performed decently well, but one thing that we wanted to do that we couldn't really, do very easily in this traditional machine learning approach is transfer learning. Okay, and what I mean by that is that, there's a lot of public single cell data sets out there. And let's say that you are doing an experiment on the brain, and let's say that you want to be cheap and you only sequence 5,000 cells and not very high coverage. Then your data is probably not very good and you probably can't get the fine, more detailed cell types. But what can you learn from these million plus brain cells that are already in the public domain? Can they help you in getting a better estimate of your underlying lambda? Or let's say that you do, you bust your wallet and you sequence 100,000 brain cells and you pretty big experiment, you're aiming for that Nature paper. And then what happens is that maybe there will be a rare cell type in your data set that it matters for the disease you're studying, and you only got a few thousand cells for that rare cell type. Would you want to look in this public domain to see if others have studied that same or came across that same cell type? So these are the questions that is very natural to ask. For example, for blood, we just last checked, there was like 700,000 blood cells in the literature, probably more. And then if there's recently been some large publications of kidney and lung, and virtually every single tissue, right? So there's multiple efforts of building human cell atlases, and within a few years, there's going to be basically millions of cells in the public domain for every single tissue you are studying. And don't forget about the mouse, right? So, there's already 2 complete mouse atlases. So for these reasons, because deep learning has had success in image processing in these kind of transfer learning tasks, we thought we will try substituting an autoencoder for this, estimating this lambda manifold. So this is a slightly more complicated autoencoder than the one I showed you earlier. It's complicated partly because we want to combine mouse and human data. And between mouse and human, there are homologous genes that are shared, and then there are those species-specific genes that are in these separate networks. And so we have this network to combine these 2 species. Okay, and And then so the human specific genes will get fed into this network. You will get a latent embedding. And then these two, the gray and the purple networks will combine to give you the human specific denoised or mean estimate. So another thing I want to say is that in fitting this network, we didn't use squared error loss because the data is counts, right? We have actually a specific error model for the data. So why not use that? So our training objective is negative binomial likelihood. So the likelihood function, log likelihood function of the negative binomial with gene-specific dispersions and cell-specific library sizes. And the gene-specific dispersions are estimated by maximum likelihood from the data. Okay, so how do we actually use this for transfer learning? So what we do is so simple, it's deceptively simple. What you do is you take this network and then you do a pre-training. You train it on these public data sets. So you go until until you want to do some early stopping and make sure that the weights stabilize. And then that is the initialization of the weights that you use for training on your target data. Okay, so in that way, the public data sets are providing you prior information for your data, but this prior information is used in a very different way than traditional Bayesian inference. Okay, so in the target data, instead of you starting from a completely random initialization, you start with the weights, the values of the weights you got from training on the public data sets. But then there are a few key details in this that actually matters a lot in terms of how successful it is. The first is not all genes are well fit by this model. So we run cross-validation to identify those genes that are poorly fit, and then we just replace the predictions by the target data mean. Okay, and then the second thing we did was, of course, this empirical-based shrinkage I told you about earlier on, where we took a weighted average of this autoencoder fit and the noisy data point. So then the question is, how much can we learn from data generated from different labs, conditions, and technologies, and can we learn across different species? Okay, so I'm going to show you two example data sets, okay? And then hopefully that will convince you that there's something here to be exploited. So in the first data set, this is the same data set I showed you in the slide earlier, is 900 blood cells. And then, the problem here is that the T cells are all mixed up and we want to be able to separate them. So the first thing we did was we just applied this autoencoder algorithm to it without any pre-training. So you only have 900 cells. Remember, this is a huge network. Okay, so you don't the data number of data points to parameters is really small. So without pre-training, can you do better than original? And actually, you are able to tease apart some of the cell types, right? So I would say this looks better than that, but not much better. If you were to pre-train on the human cell atlas, which is at the time we did it, it was 500,000 immune cells, you see that you are able to tell apart all of the T cell subtypes that were known for this data set. In this original UMAP, the pink and green are mixed together, but if you were to take these cells and do a separate UMAP of them by themselves, you see that all separate cells. And you see in the heat map, the T cell markers become much more clear. Then we ask the question, okay, so what if your target data contains a cell type that is not in the pre-training data? Okay, so what we did was we took these T regulatory cells, which is this orange cluster, and we removed all of the orange cells from the human cell atlas. Okay, so we have never seen that cell type. Then when we pre-train on a human cell atlas, are we going to bias our results to sort of not be able to tell apart the cell type? And this is the result. You see that the regulatory cells are still separable. Okay, so there's some robustness in here, and we tried this with different cell types, and it's pretty much the same. Okay.
Just a quick comment. The number on the bottom right corner of each figure, that's accuracy or? Yeah, the accuracy of the clustering. Okay. So it's just I forgot what metric we used for the paper for this accuracy. Yeah, so it's just the higher the better.
Okay. So, that was a sort of the first example. The second example is more challenging. Okay, here what we're doing is we're trying to see whether we can learn across species. So we started with high coverage human brain cells. Okay, that was collected in this paper. I think there were 2,000 of them or something like that. And then we took these cells and we downsampled them to get low coverage target data. Okay, from the original study that has high coverage data, we have the true cell types. And then what we want to do is we want to see whether we can learn from mouse brain cells to be able to take this low coverage data and recover from it the signals that were originally in the high coverage data. Okay, does that make sense? So here is the downsampled low coverage human data. Okay, you see that all the cell types are mixed together. If you were to apply the autoencoder-based denoising method to it without any pre-training, you do get slightly better cell type separation. But let's see what happens when you pre-train on either the human or the mouse data. Okay, here I have to say that both data sets are not big. It's only a few thousand cells, okay? But you see that with pre-training on a few thousand cells, even on the mouse data, you get much better clustering, okay? So this shows that it is possible with the neural network to borrow information across species. Now, one worry we had in here is, does transfer learning from mouse erase human-specific patterns? Again, because do we erase those genes that have human-specific expression patterns? So what do we mean by this? So let's say, we started with, high coverage human data. That's what we started with. And what we did was we downsampled to get low coverage human data. And then we had mouse data that was pretty good quality that of the same tissue. And what we did was we used the mouse data to pre-train an autoencoder. And then we used that pre-trained weights as initialization to denoise the human data. Now, what do I mean by erasing human-specific patterns? Well, what we could do is we can find the differentially expressed genes between the original high-coverage human data and the mouse data and compute their log fold change between species. And then we can also do the same thing between our denoised human data and the mouse data. Now, the question is, do these log fold changes look the same? If they do, then good. That means that we have preserved the human-specific gene expression. So we looked at the 5 dominant cell types. And then what's plotted on the x-axis is the log fold change from the original data, and the y-axis is log fold change from the denoised data. And you see that indeed they mostly lie along the identity line, which is reassuring. Now, why is that so? Okay, so how do we guard against transfer bias? Well, there are 3 things that we did that we thought were important. First is that we did a 2-stage weight training. Obviously, you have to fine-tune the weights again on the human data after you train a mouse. That is a no-brainer. But then there's also that cross-validation step I told you about, and the empirical-based weighting step that follows. Now, if you were to remove those 2 steps from the algorithm, you see that the plots don't look as great. In fact, there is a enrichment of points that are sort of at the x-axis, which is basically sort of a dampening of the differential expression signal. Okay, so it is important when you do transfer learning. The devil is in the details. You don't want to introduce bias. Okay, so I think maybe we should have a break. And then I will talk about batch alignment and data integration. And I will try to, if there's any questions, you could.
Yeah, I think there's one question I just try to answer. When you map from human to mouse, it's possible, right? There cannot be one-to-one map. I'm not sure whether you need to remove some genes that exist in human but not in mouse.
Yeah. So right, that's a good question. So can you
repeat the question? It wasn't audible to me.
Yeah, so the question is, when we do the transfer learning from human to mouse, the genome are not exactly the same. There may be some genes in human, but does not have exact match in mouse. So in that situation, how to handle that? Yeah.
Right. Yeah. So that's a tricky part, and I think we tried this autoencoder architecture. I don't think it's the best one, and I think there could be a lot of research in thinking about how to do this. So between human and mouse, there are these homologous genes, which are these purple nodes in the middle. Those are the genes that they share, and then each species actually has their own genes that are not in the other species. So when we pre-train the mouse, we sort of pre-train this bottom and middle part of the network. And then this bottom part, which is just mouse-specific genes, they are no longer, they're not useful for transfer learning. So what it contributes is this middle network. So the weights here are initialized, are basically carried over to the human. And then when you train again in the human, these weights are randomly initialized.
I have a question. So like when you talk about transfer learning, how much like these studies should be like similar? Like for example, if I do 2 clinical trials, one like in US, one in China, like would that be on the same medication? Is that can we use like transfer learning on these 2 studies, or what is?
It's very commonly done, transfer learning to integrate data from very different from different labs and very different experimental protocols. That's actually one of the main motivations of transfer learning. And I think after the break, I'll show you a study where they develop a method where you can actually integrate these data sets.
Okay.
Is that what you're asking?
Yeah, I want to see like, yeah. Right. That's something.
Yeah, and I think it's also related to another question from Megan. What's the metrics measure? How much has been transferred? I think we can probably cover this in a little bit depth later. But and this is a related question. It's I do not have exact answer. I mean, Nancy, you can correct me. But there are some examples. Like here, Nancy shows human and mouse are very different, but the signal probably is cell type difference. So that cell type difference is bigger than the difference between species. So you can still transfer that, and then transfer learning is very popular in the imaging field. You just use deep learning to learn the image, cars and cats and dogs, and then you can apply it to medical imaging to look at the whole slice image, to look at cells, the tumors. So yeah, it's depends on the problem, but I think many examples shows you can transfer learning many different situations.
Yeah, as to how much information we've thought about this, but we don't actually have a good way to really quantify it. But what we found is that we have yet to find a situation where transfer learning hurts. It usually helps somewhat, because it's always better than a random initialization. Right? There's always something that is shared between these 2 data sets. That's better than the random initialization.
I missed a technical point. In the previous slide, you talked about using the empirical Bayes. Can you explain again how that's used?
Yeah, so I think it's this slide, right?
You had it on multiple slides.
Okay.
Just conceptually, how is the empirical Bayes?
Yeah, so empirical Bayes, I mean, so we assume that, so basically in this model, you have to have a model, right? Here we have basically a very simple model where, lambda, X is derived from lambda and Y is derived from X. In the distribution of X given lambda, there is one parameter that controls how much dispersion there is, and that relies on a gene. So it's that's a gene-specific parameter, theta G. In the distribution that controls how Y comes from X, there is one parameter, LC, which we know. That's the cell-specific library size. So there's a cell-specific library size parameter, and there's a gene-specific dispersion parameter. So it turns out that this is your estimate your, if you assume that this is a gamma distribution, right, then this is a negative binomial, and then this would be the maximum likelihood estimate of x hat, right? If you don't assume a gamma distribution, then this is the minimum variance estimate. It's just the weighted average of YGC over actually this should be LC. This is a typo, LC plus 1 minus 7. So this is just a weighted average of your observed raw data point and your machine learning fit. So and I didn't write out the terms how this the exact forms of the W, but it depends on these three quantities as you intuitively expect it to. Does that make sense? So why is it empirical based? Well, you estimate data by putting information across cells for a gene, and then you estimate LC by putting information across genes across cells. So that's how you sort of pull information to estimate these parameters.
I see. So the empirical base prior is kind of fed into the individual gene estimates as a prior. Yeah, perfect. Thank you.
Okay. So maybe we should continue. We have about 20 minutes, so I think I could go over these other aspects of transfer learning. So, transfer learning is actually a often misused term, and I shouldn't say misuse because people have different interpretations of what transfer learning is. Some papers use transfer learning to mean data integration, integrating data from multiple batches or multiple species or multiple, cohorts of patients. Okay, so let's talk about batch alignment and data integration. So remember, what is this problem again? This is a problem where you start with supposedly a homogeneous population of cells. You do two experiments, supposedly the same experiments on them, and you get data that is systematically different from each other. Okay, and then in addressing this problem, what we did was this is a work in my lab, which is very similar to the next paper that I'll show you from, which was recently published. We all use a variational autoencoder for this problem. And what we did was we added nodes for batch. So this is a one-hot encoding of the batch. So let's say you have 3 batches, then you have 3, this B will be 3 nodes, 1, 0, 0, 0, 1, 0, or 0, 0, 1. So it's a one-hot encoding. We have that node going into the encoder as well as going into the decoder. And you would think that adding this node, with a variational autoencoder, you will automatically get Z values that are orthogonal to it. That's not guaranteed, okay? Because there's nothing in the criterion function that says that B needs to be orthogonal to Z. In fact, and this is an area of research, a lot of recent research, is that if you increase this KL penalty term, you put a beta in front of it and increase that beta, if you weight the KL penalty term really heavily, that leads to what's called a disentanglement variational autoencoder, and that makes the Z values orthogonal. And we've tried that, and that magically makes it orthogonal to B either, but it's not sure for sure. So what we did was we experimented with adding this what's called a batch reconstruction loss here. This is the alpha. Alpha is a weight that we can adjust. And what this batch reconstruction loss does is it says that if you set Z to 0, means I don't want to use any information from the middle layer encoding, and just use the B value, I want to be able to reconstruct Y as well as possible. So that is basically fitting a batch specific. You want everything that is batch specific to go into the weights for B. And what is remaining will go into the weights for Z. That was the intuition. So if we try this with a single layer encoder and single layer decoder and a linear activation function, and this loss term, adding this loss term is equivalent to conditional PCA. So you might imagine, right? What if you want to do a PCA and you want to get an embedding that is orthogonal to a covariate? What you would do is you would first regress your Y on the covariate, get the residuals, and do PCA on that. That is essentially what's called conditional PCA. So if you were to have a simple linear encoder and decoder and have this term, you get conditional PCA. So with a nonlinearity, we're not sure what exactly is happening, but it basically, empirically in all our single cell data, this allows you to get Z that is orthogonal to B. Okay, so let's look at some experiments on data. And here I'm showing you a data set with 3 blood cell types, the monocytes, the T and NK cells, and the B cells. They're the different colors on the top row, and 3 protocols, which are the bottom row. They're just different ways of doing single cell experiments. And you see that the leftmost plot is, these are all UMAP plots. It shows you the original data. And you know, you see that how bad the batch effect is. The purple and green and orange cells are all very separate from each other, right? So you don't know whether a cluster is due to a batch or due to a cell type. If you apply this method that I showed you before, which we call SaberCat, it actually merges the clusters together very well. Okay, so we tried both the counts and the estimates. Estimates are the X hats. Estimates are the lambda values. The counts are the X hats. And they're pretty much the same. And then we compared it to 2 existing methods, Seurat and SCVI. What I want to note is that SCVI is almost exactly like our method. It uses a variational autoencoder, but it doesn't have this batch reconstruction loss term. So what that does is that it does pretty well in aligning the data in low dimensional space. But if you decode it back to high dimensional space, it doesn't remove all of the batch effects. Okay, so here we have the Kruskal-Wallis p-value for distribution difference between batch within each cell type. So within each of the monocytes and the B cells and the T cells, how different are the batches? And the p-value we want that to be as big as possible, right? We don't want it to be small. So you see that without the batch correction term, it doesn't do as well as with the batch correction term. And here is just an example of one gene. You see that in the original data, there's a lot of batch differences between the 3 colors. And then when you align it, you get very homogeneous distributions between the 3 batches. Okay, so I want to show you sort of like the grand plan of how this could be, which is in this Nature Biotech paper that was published last year. So what they say is that when you want to do transfer learning, when they say transfer learning, what they mean is data alignment, data integration. You want to integrate data across studies, across organs, across species, and across healthy and disease states. Okay, so how does that work? So it says that, well, there's a lot of buildup of public single cell data, right? So you have many different studies for, say, a given organ like the brain. And then you would fit a variational autoencoder on that. And then, this is exactly like the same as the model I showed you earlier. Everybody had the same idea. And then, that would give you a model, okay, for brain. The weights for the blue nodes are sort of brain specific, and the pink nodes just correspond to batch. Now, let's say that you have your own study now, the query data. And then what you would do is you would download the model for the brain. You would go into our website and download the model for the brain, get the weights for that, and then you would put this adapter. This adapter is basically this node that you add here with the weights and the edges that are specific to your study. And then you would add the adapter to both the input and to the encoder and the decoder. And then you would train this. You would basically update the weights, but only of the adapter. So that's where they're different. They say, okay, let's not touch the downloaded blue weights. We only want to train the weights of the adapter. So what they want to do is they have this reference data and they want to train the adapter to sort of merge your query data to the reference data. Okay. It's like, and then you will get different adapters corresponding to different studies, different batches, different species, maybe, maybe the data you downloaded is of the mouse and you have human, you have a human specific adapter. this is as plug and play as it gets in terms of modeling, right? So, so the question is, does this work? Okay, so they demonstrate this on quite a number of different data sets in the paper. Here is, I think, a good demonstration on Pancreas data. So here is the, okay, so the data actually has 5 different studies, okay, 5 different batches, okay, using different technology. And what they did was they used 3 of those batches as what they call the reference. Reference being what the pink data sets. They train those as a reference, and then, and then they are able to align the reference. You see how the reference data, the 3 different reference data sets are all separate. That's a batch effect. And then after you train it, you get these basically the cell types aligning together. And then, that's good. Okay, so then what they did was they took this SS2 study. This is a new study. And what's more is that they removed these as alpha cells from the reference. So they say, oh, well, let's look at a case where my new study has a new unseen cell type that was not in the reference. So that's the unseen cell type right here. So you see that when they add the SS2, they are able to sort of integrate the SS2 to the reference data. Okay, so they lie on top of each other now, right? The gray and the green. And then also, but then they didn't, it's like the unseen cell type stood out on its own, which it should, because it shouldn't get aligned to the only the seen cell types. And same thing with the other study that they left out. So this was a proof of concept that basically with a variational autoencoder, you can use it to sort of do data integration, and naturally new signals, new axes of variation in the data set you have are not going to be forced to merge with existing axes of variation. So obviously, this is only last year, and there's a lot of labs still working on this problem. And I think there's a lot more to be done. So that, with time, we'll see what kind of architectures, what kind of training methods would work best. Okay, now here is another very exciting application of variational autoencoders, inferring unseen cell states. What we mean by that is that, let's say that you have in your lab, maybe 4 different cell types, and they are sitting at a resting state. Let's say that you did experiments that perturbed them in some way, and then you're able to observe that for the 3 cell types, but you didn't do the experiment for the blue cell type. You want to know what happens to that cell type if you were to stimulate it. Okay, this is a very relevant question, because sometimes imagine that these, the purple cell type and the brown cell type is the mouse. And the mouse, you can do stuff with the mouse. You can give it a drug, right? But the blue cell type is human. So you want to know what happens to the human cell type, equivalent of the mouse cell type, if you were to treat it with the same drug. You can't do this with humans for ethical reasons. But you can imagine many scenarios where you want to predict what happens to a cell type in an experiment that you cannot do. So how does this work? So, if you were to just visualize these cell types in high dimensional space, you'll be able to see how these different cell types change. But, it's hard to sort of take these vectors, do an average or whatever, and put them on the blue cell. That's not going to work very well. So what they did was they encoded these cell types into a latent space using a variational autoencoder. And in the latent space, they estimated they took the difference between the means of these cell type clusters to get this delta, which is what they say is the effect of the perturbation. And then you basically add that delta to the blue cell types in the latent space, and then decode it, and then decode it back. And that gives you what the blue cell types should look like if you were to apply the same perturbation. It all seems like magic. I mean, there's a lot of assumptions to this, right? You are assuming that in the latent space, the purple and these different cell types have the same delta vector. That's a big assumption. And so, but then they showed examples in their paper where it seems to actually work pretty well. So here's an example of a sort of immune cells on which they applied interferon gamma stimulation. So interferon gamma is a commonly cell surface receptor. It is a way that immune cells receive inflammation signals. And here's a UMAP embedding, where at the top you see whether it is stimulated versus unstimulated, right? So all the stimulated cells are at the top. And then at the bottom are the cell types, right? The B cells, the T cells, etc. And what they did was they held out a cell type during training, and then they try to see whether they can recover the stimulation state of the cell type. And then, so let's look at how it works. So here you have, basically it's divided into B cells, T cells, different kinds of T cells, monocytes, NK cells, and then each column is a cell type, it's like a gene, okay? And then there are these cell type specific markers that are uniform. These are the uniform interferon gamma markers. And then there are these cell type specific interferon gamma markers. And you see that basically the prediction and the prediction tracks the real, right? So it seems as if the prediction is pretty good. I mean, given that, this is such a lofty goal, it's amazing that they can go so far. Okay, I'm pretty sure that this doesn't work for every data set, but the fact that they were able to work for some data sets in their paper, I think is pretty amazing.
Does this require that, can I ask a quick question?
Yeah.
Does this require that the differential, like the signal from interferon gamma will be similar across cell types, basically? That's what you were saying.
They must be similar across cell types in the latent space. Okay, and that's why the latent space is critical. I don't know why they should be similar across cell types in the latent space. I'm sure there's a lot of caveats.
Yeah, very interesting. Thanks.
Okay, so there's a lot of things to mull over, but let's just pause here and maybe have a summary and a discussion perhaps of why deep learning has been successful for single cell transcriptomic data. Okay, because there's plenty of deep learning papers in single cell transcriptomics. There's lots of interest in this. Well, one reason is that cell transcriptomes are tightly constrained high dimensional vectors, in a sense that they don't, they are, you have 20,000 genes, you map them out, they are tightly constrained. They have these weird high dimensional geometries because of the underlying pathways and processes that these genes coordinate as part of to define the cell. Okay, so basically a cell is defined by these latent pathways that are happening inside it. And these latent pathways constrain the genes to coordinate them to be expressed in a coordinated fashion. Okay, and here's something that I'm starting to realize is that a cell's transcriptome pretty much defines its state, in a sense that if you can see the cell's transcriptome, you know pretty much a lot about the cell. I'm working with my collaborator is a protein biologist, and he works from proteomics, and I think we might disagree, but we have been looking at, for example, multi-omic data that pairs RNA with protein, so site-seq data where you collect protein markers as well as the RNA of a transcriptome of cell, or ATAC and RNA. And what we see is that when we have these multi-omic data, the new information that is added by these other omics dimensions, there's some, but not a whole lot. So what is that saying is that if you see the RNA transcriptome of a cell, you're seeing 80% of what the cell is. So, compare that to image processing, right? If you see a picture of a cat, it's a cat. If you see a picture of a dog, there's no picture, unless it's fuzzed up, there's no ambiguity of what you're seeing. Right, so that's why images are nice things to learn from for neural networks. Compare this to the other extreme where you're looking at a GWAS study, right? You can know the genome of this person, but there's still a lot of environmental factors that come in to determine its phenotype. So that is a case where there's a low signal to noise ratio. In single cell data, single cell transcriptomics pretty much defines the state. Okay, so it's a high dimensional, highly featureizable vector with complex dependencies between genes that pretty much defines the cell state. Data sets in single cell field are very large and heterogeneous. You're going to have millions and millions of cells, and they're very heterogeneous. So there's a lot of interest in learning across data sets. And neural networks makes this learning easy. So we saw 3 examples of transfer learning through neural networks. The first is through weight initialization. That's the easiest. The second is through a conditional variational autoencoder and these things that are called adapter nodes. And then the third is to basically doing linear operations in the latent space. And we see that they all have some degree of success in this field. Okay, so I think, why don't we stop here? Maybe I would just take the next 15 minutes in the next lecture to go through this example. This is I think a beautiful application of variational autoencoders to looking at protein sequences. And we can save that for the next time.
Yeah, thank you, Nancy. I think we exactly on time now. So
yeah,
we probably can stop here. Stop recording.
Recording stopped.
Thank you, everyone.
yeah.
See you
Thank you.
next Tuesday. It's still the same Zoom link. Some has
Yeah. And I think that I had a question of where the videos are. We're going to share. We're uploading them to YouTube through a private channel, and they will be shared.
Yeah. We'll send an email on the link of the video. Okay. All right. Thank you.
Okay. See you guys next week.
Yeah. Bye.